screen is not visible.
Not visible? I think someone said they were able to see it. Okay, share the screen. Now?
No.
Now is it visible?
No, ma'am.
Okay. Let me check for a minute.
It is visible, ma'am.
Yeah, is it visible now?
Yes, ma'am. It is visible.
Yes, madam.
Okay, yeah, so we'll start with our class. Right, so we are going to talk about stem cells today because our whole topic is about animal biotechnology, and without understanding the stem cells, it would be a little bit difficult for everyone to understand. So, though it's going to be a recap of what we did when we were talking about it in offline class, nevertheless, it's going to be again interesting for those who have listened and for those who haven't listened to this class. So we'll talk about stem cells, and we all know what stem cells are and how important it is in our regenerative medicine, and also when you're going to talk about different diseases. Maybe it can be autoimmune disease or a genetic disease. Scientists are really working hard towards developing stem cells so that they can cure different types of diseases. So, on those basis, we'll start with what exactly is stem cells and from where we can get these stem cells. So, if you're going to have any doubts, you can just interrupt me in between so that I will again explain to you all what exactly it is. Is that clear, students?
Yes, ma'am.
Okay. So yeah, if you can see in this picture, a totipotent cell. So that's a totipotent cell, and that totipotent cell, after the cell division, they're going to form into embryonic stem cells, which slowly tries to become a pluripotent stem cells, because from a toti, it's going to become a pluripotent. The pluripotent stem cells is going to give rise to three different germ layers, that is the endoderm, mesoderm, and the ectoderm, which later on becomes a multipotent stem cells. And from the multipotent stem cells, we can generate different types of cells which can become oligo or also nullipotent. That is, nullipotent is when it comes to the final stage of what that particular cell is destined to be. So these are the different types of cells we'll be talking about. So remember, we'll be talking about totipotent, we'll be talking about pluripotent, multipotent, and oligo and nullipotent stem cells. So what exactly is stem cells? So these are special human cells that are able to develop into many different cell types. That is, they can form into whatever type of cells that you want your cell or organ that has to develop. So from muscles to brain to liver cells to kidney cells, you name it, and they're going to form, because these are the cells that is going to give rise to a complete human being. So in some case, they can also fix damaged tissues and may also be used in one day. This can also be used to serious Alzheimer diseases and paralysis, as I was talking about. So this is the importance of stem cells. So these are of two types, that is embryonic stem cells and adult stem
Ma'am,
cells.
voice is
So
not
under
clear.
embryonic stem cells, uh, okay, right now, how about now? Is it clear?
Yes, ma'am.
Yeah. So when you're talking about embryonic stem cells, this is the latest research that is going on. Though there is a lot of controversy behind those embryonic stem cells, because what is embryonic stem cells? The cells that have been taken out from a fertilized egg. So that's called as embryonic stem cells. Though we will be able to produce different types of cells, because remember when we are talking about this picture over there, so this embryonic stem cell is a totipotent and also a pluripotent stem cell. So this is going to form the complete human, the adult human species, or it can be any species that it's going to form. So this embryonic stem cells is pluripotent, meaning that they can turn into more than one type of cells. That's the importance of embryonic stem cells. So other than that, we also have adult stem cells. As I told you all, embryonic stem cells, a little bit controversy, because people think that when you're going to take out the cells from the embryo, we are trying to kill a life. So thereby there is a lot of controversy behind that, though it's a very beautiful cell, wherein you can grow into whatever cell that you want to. Then you also have an alternative that's called as adult stem cells. So you're talking about adult stem cells. There are two types of adult stem cells. One that comes from a developed tissue, that is an adult form, when a baby is born, and up to the adult stage. The cells that are present in the humans, they are called as adult stem cells. You can call it as somatic cells also, because this can be taken out from either the brain or a skin, and also from the bone marrow. And though they are very small in number, because you don't find much of stem cells as you find in the embryonic stem cells, so they are a little bit less when compared to embryonic stem cells. But and of course, they definitely can make different types of cells that we want to induce them to be. So for example, when you're going to take out a stem cell that comes from a liver, they can only form liver cells. You can't expect a liver cell to form a brain cell or a muscle cell. So a liver cell is going to form only a liver cells. So there is a second type of stem cell. This is a very important point over here, that second type of stem cell, because this is fast growing into an important stem cells, that's called as induced pluripotent stem cells. So here we are going to induce the cells to form a pluripotent. Remember, a pluripotent stem cell is going to form different germ layers that we are talking about, that is a ecto, meso, and an endoderm. So here you're going to make the cells, the adult stem cells, to induce to form into a pluripotent stem cells. So these are going to be done in a laboratory, because they need a lot of cells, some different type of chemicals that has to be induced them to be forming into a pluripotent. And scientists first reported that human stem cell could be changed in this way in 2006. So this is a very, very new type of stem cells that's coming up. And this induced pluripotent stem cells don't seem to differ from the embryonic stem cell. That is one more important point. They are not so very different from the embryonic stem cell. That's why I said this is the one subject, one type of research where it is growing like very vast. So if you all are thinking of doing some research in your future, you can definitely take up at this place where you can induce the cells to form pluripotent stem cells. Though of course scientists are yet to find one cell that can develop into different type, that is embryonic stem cells. So we'll talk a little bit about a history of human embryonic stem cells. So in 1998, James Thomson isolated the cells from the inner cell mass. So you can see that that's the inner cell mass of the embryo over there, and developed the first human embryonic stem cells. So this all started in 1998. So from there on, there was a little bit of hope that one fine day we humans are going to find a solution for any type of diseases, or also you can grow a new limb or a new lost organ. We can say a regeneration probably. So there was a hope kind of in 1998. And also in the same year, John Gerhardt derived human embryonic germ cells from the cells in the fetal gonadal tissue. Understand the word fetal gonadal tissue. That is, from the fetus, the gonad, that is the reproductive tissues, was isolated by John Gerhardt in 1998. Fetus itself, such a small, tiny living organisms that is still growing, and from that they were able to take out the gonadal tissue. So fetal gonadal tissue, that's the primordial germ cells that we'll be talking about. And pluripotent stem cell lines were developed from both these sources, that is from early embryo and also from the fetal gonadal tissue. Then in 1999, first successful human transplant of insulin making cells from cadavers was done. Cadavers, that's a dead bodies. So they were able to take out the cells and they were able to transplant for insulin making, definitely for diabetes. And in 2001, first cloned human embryo, that's only up till 6-cell stage, created by advanced cell technology. And 2004, Harvard researchers grew stem cells from the embryos. So slowly, day by day, technology has been increased, and you can see different formats of here. So this is what the history of somatic cell nuclear transfer. If you remember, you're going to talk about the cloning. When you're going to talk about the cloning is not a fertilization between egg and a sperm, but you're going to grow a cell from the cell itself, that is from the egg. You're going to take out the somatic cell nucleus and you're going to fuse it and you're going to start to grow them. So that's called a somatic cell nuclear transfer. Somatic, you remember, it's not the reproductive organ, but a cell organ, that is a tissue organs over there. So in 1952, Briggs and Kring cloned tadpoles. The first one to be cloned was a tadpole. And then in 1996, as we all know, the famous Dolly the sheep was cloned. And then later on followed by mice, cows, and pigs were cloned following. And there's a lot more to come. Yeah, so in 2001 to 2005, cats were cloned, rabbits, mule, bull, and also a dog was cloned. Cloning, we all know, that's a Xerox copy of the parent. So that's called as cloning. So all these were done up till 2005 and also still going on, a lot more to come up. So we'll talk about what stem cells exactly are. So these are undifferentiated cells capable of cell renew and to differentiate into different cell types or tissue during embryonic development and throughout adulthood. So what do you understand by this first word? Undifferentiated cells capable of self-renew. That is, the one set, the undifferentiated cell is going to divide into two types of cell. One self-renew and the other one is going to differentiate into the different types of tissue during embryonic development. Self-renew is a stock over there, and that stock is going to be utilized throughout the adulthood, where whenever you want a stem cell or whenever you want a DNA repair to occur, whenever you want a tissue repair to occur, these are the cells that is going to come back to produce that different type of cells. So it is undifferentiated. So for example, you can also take an idea of a clay. So you have a clay over there, and that clay can be molded into whatever type you want. So for example, you want to make a statue, you want to make an animal model, you want to make a bird, so you can make them into different type. So that's called as undifferentiated cells, which are capable of self-renewing. So they have the possibility to become specialized cell, and they have the ability to divide continuously and develop into various other kinds of cells. So for example, remember that we need a continuous supply of RBCs and WBCs. So they have the ability to divide continuously. And immune potential, because it can help to treat wide range of medical problems and discover known as regenerative medicine. Remember, in the first year we were talking about regeneration, wherein we talked about earthworm model over there. So this stem cell discovery has made a whole lot of new technology, that is the regenerative medicines. So there's a stem cell timeline. So you cannot expect the stem cell to be the same throughout the developing year, because when you're going to talk about totipotent, pluripotent, they have a limited timeline. So what is this limited timeline? When a zygote is formed, the first one over there, when a zygote is formed, this is a zygote, and this zygote is called as a totipotent cell. Toti means it is going to form into different type of forms of cells. That is, they can either become an embryo, they can also become an extra embryonic stem cells. So that is called as totipotent. Understand? Zygote is going to form into an embryo, and also it is going to form an extra embryonic stem cells. That is, extra embryonic is the placenta, the umbilical cord, the covering, the amnion, the amniotic fluid, and all those cells that are going to be developed. That's called as totipotent stem cells. That is going to stay only for say from day 1 to day 3. So that's a timeline for totipotent. Day 1 to day 3 is the timeline for totipotent. And then once it goes into day 4, day 5 to day 7, so for example, from day 5 to day 7, they are going to form into pluripotent stem cells. So here, pluripotent means the cell is going to form only either an embryo or the trophectodome or the trophosis cells that are going to form, that's going to form the extra embryonic cell. They are differentiated over here, either extra embryonic stem cells or embryonic stem cells over there. That's called as pluripotent stem cells. Then comes the multipotent stem cells. After pluripotent, it's a multipotent. Here, the multipotent stem cell can be found out from cord blood cells and also from the placental cells. So you can take, for example, you want to make some multi. Here, understand, multi is not a toti. It has reduced over there. So multi means few number of differentiated cells can be formed from cord blood cells and also from the placental cells. So that's why nowadays we have come across with a lot of cord blood banks, where once the child is born, within 15 minutes the blood is collected and this blood is stored as cord blood cells in the banks over there, so that whenever the child needs its own cell for growth of different cell types, this can be used. In the same line, the placental stem cells can also be taken out from that, so that you can do some research work. So that is multipotent stem cells. Then once the child is born, now the cells have become either multipotent or it's also called as adult stem cells. So this can be made as adult stem cells or multipotent stem cells from infant to adult. So that is a stem cell timeline. Now here again, we can, in this picture, it will be very easy to understand now what exactly I was talking about a totipotent. So once the egg is fertilized, the zygote that is formed is going to be called as totipotent stem cells over there. And this, once it is going to divide itself, it is going to form into the next cell, that is the blastome. So the inner mast cell, so this is the inner mast cells, the purple color cells. So this is the cell that is going to form the future embryo, and that is the trophectodome cells, which is going to form the other different extra embryonic stem cells for the developing embryo over there. So once you can isolate the cells from the blastodome, and if you're going to culture them in vitro culture by adding different types of growth medium, you're going to culture them into whatever cell culture that you want them to grow as. Otherwise, these stem cells are going to form into hematopoietic, neuronal stem cells and mesenchymal stem cells. Now, when you're going to talk about how the different stages going to be, you can just remember this particular pic over there. So that's a zygote, which is a totipotent. An embryonic stem cell is called as pluripotent. The germ cell layers, that is the ectoderm, mesodome, endodome, that's called as a multipotent stem cell. And from the multipotent stem cells, you're going to form into an oligopotent stem cells. What's an oligopotent stem cells? An oligopotent stem cells is, again, they are defined over there, they are restricted, but one cell can give rise to different types of cells. That is only 3 different types of cells. So what are they? So when you're going to talk about the hematopoietic stem cells, when you're talking about hematopoietic stem cells, if you're going to talk about the leukocytes, and also the myeloid cells, which are going to form into WBCs. One WBC cell can either become a neutrophil, they can become a basophil or a eosinophil. So that is oligopotent stem cells. Then the next, it is again, if you can see in this picture, it is going to get restricted as the timeline is increasing over there. So here from the oligopotent, they are going to form a tissue determined stem cells, that's called as a tri or bipotent. They are either 3 or 2 different types of cells can be formed over there. And once the cells is differentiated into the destined cell that it has to be, then that cell becomes a nullipotent. What do you mean by nullipotent? There is no more cell division that is going to form into a different type of cell. So it has reached its target cell, what it has to be formed as. So for example, a neuronal cell has become a neuron, a liver cell has become a liver cell, hepatocyte cells, and a bone, the osteoblast, is going to form into the particular bone cell. So there is no more further division of that particular destined cell that it has been formed. That's called as terminal cell or nullipotent stem cells. So we'll talk about each and every type of cell in detail over here now. So when they ask you to talk about the different types of stem cells, you're going to talk about these 5 different types of stem cells. That is the totipotent, pluripotent, multipotent, oligo, and unipotent. You might be asked for a major question over there. So describe stem cells and the different types of stem cells that you come across. So you're going to talk about this. So when you're going to talk about totipotent or omni is all varieties of cells that can be formed. That's called as a totipotent stem cells. So this can be differentiated into embryonic as well as extra embryonic tissue. So what is an extra embryonic tissue over here? It can be a chorion, it can be a yolk sac, it can be an amnion, and also it can be an allantois. And in humans, this can be formed as a placenta. Why? Because the development of the embryo needs the placenta. If you remember, when we were talking about the chick embryo, in chick embryo we were talking about the yolk sac, we were talking about the amnion, the allantois. Allantois, if you remember, in the 96R embryo, we were talking about a small bag that is arising, the allantois, where the excreta has been collected in that bag. So not only the development of the chick, but also the extra embryonic cells that has to be formed is formed by this totipotent stem cell. So when you're talking about the humans and other major mammals, here you're going to talk about the embryo as well as the placenta that is going to be attached with the mother and the child over there. So the umbilical cord and all those, which is going to be in connection with the growth of the embryo over there. So that's called as totipotent stem cells. So here we're going to talk about embryonic as well as the extra embryonic tissue cells that are going to be formed. So that's called as totipotent or the entire stem cells. And the most important characteristic of this totipotent stem cells is they're going to generate a fully functional living organism. So you should be really intrigued by how a particular single cell is going to form a complete living organism. So when we are talking about the developmental biology, if you remember, we were talking about how from a single cell, a complete living organism is going to be generated. So the best known example of totipotent is, of course, the fertilized egg. So fertilized egg is going to form the different types of cells. So the example, when I ask you what is an example of a totipotent stem cell, blindly you can say it's the fertilized egg that is going to be a totipotent stem cell. And it is around 4 days post fertilization that these cells begin to form pluripotent. So they have a very short life timeline, that is from day 1 to day 4 they are going to become from totipotent to pluripotent. That is the next type of cells. So what's this next type of cells? The pluripotent stem cells over here, if you can see, this can self-renew, and also they can differentiate it into 3 germ layers, that is the ectoderm, the mesoderm, and the endodermal germ layers. And these further differentiate into different forms of tissue and organs within the human organisms. That is, if you remember, what are the tissue, what are the organs that are going to form from the ectoderm? We're going to talk about the skin and also the nervous system. When you're going to talk about the mesoderm, you're going to talk about the development of the muscles. And then when you're going to talk about the endoderm, you're going to talk about the GI tract. So these are the different examples of different organs that are going to be formed from this particular germ layers. There are several known types of pluripotent stem cells. Among them, the natural pluripotent stem cells is the embryonic stem cells, which are the best example. And the embryonic stem cells are unique cells that exist in an early stage of the embryo. Again, the timeline over there is from 5 days to say 10 or 15 days or so, not more than that. So another type of human-made pluripotent stem cells also exists, which is called as induced pluripotent stem cells. Remember, I was talking about this induced pluripotent stem cells when I was talking about the first two slides over there. The iPS cells, which were first produced from the mouse cells in 2006 and in human cells in 2007. And as the name indicates, here when you're talking about induced pluripotent stem cells, you are inducing the cells to form pluripotent once again. Otherwise, they are adult cells. You're going to take out some somatic cells and you're going to induce them again to form pluripotent. That is, you're going to go back in the timeline. You want to go back in the timeline. That is, once the cell has been formed, you want them to again become a pluripotent. That is called as induced pluripotent stem cell. And as I said, this is growing very fast over there. And this is the one people prefer nowadays. People, in a sense, the ethical committee prefer this because you're not going to use the embryos to form the stem cells over there. So induced pluripotent stem cells is the next big thing that is going to occur in our future, because they are going to be functionally similar to embryonic stem cell. That's why they are very much in importance. Now, because of the ability to differentiate into wide diversity of tissue and a non-controversial nature, induced pluripotent stem cells are well suited for cellular therapy and regenerative medicine. So if you can see in this picture, you can take any adult stem cells, you can isolate any adult stem cells. In that adult stem cells, you're going to induce the iPS. That is, there are some factors that you can add to them. That is, let me tell, this called MIC, MYC, MIC, COX. Then you have ELF factors over there. There are different types of chemicals that are going to be used for them, so that when you use this, you're going to induce them to form the iPS over there, or the induced pluripotent stem cells. And here you can see, again, induced pluripotent stem cell can be changed into mesoderm, endoderm, or ectoderm. Remember, once it is an adult stem cells, you cannot change them to any stem cells. That is, they have reduced its capability. But here you're going to take it out, you're going to induce them to form pluripotent, wherein the pluripotent is going to give rise to three different germ layers. It can be meso, endo, or ecto. From there, you can bring about any type of cells that you want them to form. Okay. So before going into next step, I want you all to have a look at this great scientist, Shinya Yamanaka, the Japanese scientist and a Nobel Prize laureate. He is the one who had pioneered this induced pluripotent stem cells, and Sir was also awarded in 2012 with a Nobel Prize along with Sir John Gurdon for the discovery of mature cells which can be reprogrammed to become pluripotent, Shinya Yamanaka. So when you're going to talk about the induced pluripotent stem cell, what exactly is that we are going to talk about in this regenerative medicines using induced pluripotent stem cells. So here from a patient, you can take out the somatic cells from the patient, you can culture them. So these are the factors that I was talking about, growth factors that I was talking about, OCT4, SOX factors, growth factors that you can use into the somatic cells, and you can induce them to form into different types of stem cells. And with the help of a little bit of gene editing, you can correct the iPS, and then you can directly induce them into the healthy cells, and these cells can be replaced into the patient, where cell it's called as cell replacement therapy. In the other instance, you can also do an in vitro differentiation, that is with the help of developmental biology, with the help of genomics. You can reverse the cells, that is you can remove the problematic cell over there, and then you can induce a normal cell in that, and then you're going to add it to the patient for the drug development over there, which will help the patient to recover very fast. So these are two different model: disease modeling and cell therapy, where you can use induced pluripotent stem cells. Now, coming to multipotent stem cells. So from totipotent to pluripotent, now we are going to talk about multipotent stem cells. The multipotent stem cells can self-renew and differentiate into a specific range of cell types. So an excellent example of this cell type is the mesenchymal stem cells and the hematopoietic stem cells. So the mesenchymal stem cell can differentiate into osteoblast, monocytes, adipocytes, and chondriocytes. And these cell types are fairly diverse in their characteristics, which is why mesenchymal stem cells are classified as multipotent. So here you have to differentiate. The multipotent stem cell is not equivalent to pluripotent. Here they are going to get restricted themselves to form into only some type of stem cell. That's called as multipotent stem cell. So if you're going to take a mesenchymal stem cell and you're going to let them grow in a surface where you're going to give them the growth factors over there, you can make them into vascular endothelial cells. You can also grow them as adipose. You can also grow them as muscle tendons. Wherein this can be used for osteoarthritis patients, where the cartilage bone, or also you can use it for any accidental fractures over there. And also you can grow a new bone, a new nerves, and a muscles. So this is where the mesenchymal stem cells come into play. And when you're going to talk about hematopoietic stem cells, I'm sure everybody are aware of what is hematopoietic stem cells and from where you can get these hematopoietic stem cells, and what they are going to differentiate into. So they are going to differentiate into myeloid cells and the lymphoid cells, and this myeloid is going to give rise to the megakaryocyte, which also forms the thrombocytes or the platelets. It can also give rise to RBCs. It can also give rise to mast cells, that is the macrophagic cells, which is going to remove the microorganisms, and also the myoblasts, the agranulocytes and the granulocytes over there, which are going to give rise to basophils, neutrophils, eosinophils, and the monocytes. The monocytes, until they are in the blood circulation, they are going to be a monocyte. Once they have to go and repair a tissue, they have to squeeze out of the blood capillaries and go into the tissue to repair, is when the monocytes is going to change into macrophagic cells over there. Then when you're going to talk about the lymphoid progenitors, lymphoid is going to form into two cells, that is large granular cells and a small lymphoid cell. The large one is going to form a natural killer, and the small is going to form the B lymphocytes and the T lymphocytes, and the B lymphocytes forms the plasma cells, which is the one that is going to form your antibodies over there. And the T lymphocytes are specific cells, which are going to be antigen specific cells. That's the T lymphocytes, where the T lymphocyte is going to go to the thymus to form the T cells over there. Now coming to the next one, that is the oligopotent cells. What's an oligopotent stem cells? These are similar to the prior category, that is a multipotent, but only difference is they have become further restricted in their capacity to differentiate. So while these cells can self-renew and differentiate, they can also do so in a limited extent. So an excellent example is the hematopoietic stem cells. That is what I was talking about when I was talking about oligopotent. That is, they can form only a limited type of stem cells, 3 or 4 different stem cells that we are talking about. So these are derived from the mesoderm that can differentiate into other blood cells, specifically the HSC, that is a hematopoietic stem cells, are oligopotent stem cells that can differentiate both into myeloid and the lymphoid. So here you can see over there, they can either turn into myeloid cells or also they can turn into lymphoid cells over there, but they are not multipotent. Myeloid cell, which induces them to become basophil, dendritic cell, eosinophils, erythrocytes, macrophage, and megakaryocytes, and of course the lymphoid cells, the B cells and the T cells, and natural killer cells. So this is the same thing that I was talking about, the myeloid and the lymphoid forming into different types of cells over there. Then the last one is the unipotent stem cells. Uni is single. The name itself indicates that it is single. So at least These are the least potent and most limited type of stem cell, and an example of this type of stem cells are muscle stem cells or nerve. So that is, when this cell is going to turn into muscle cells, then they are destined to form muscle cell. They cannot form a liver cell or a nerve cell. When a nerve cell is going to form only a nerve cell, but they are not going to form into any other different tissue cells. So while they are able to self-renew and differentiate, they can only do so in a single cell type. So they are unidirectional in their differentiation capacity. Unidirectional is only one side. They cannot deviate into other different cells. So here you can see the muscle cell going to form the bone marrows over there, M cells, the N cells which is going to form the nerve cells, the heart cells, and the P cells which are going to form into the pancreatic cells over there. So when we're talking about the stem cell division and differentiation, so this is how the division occurs over there. The symmetric stem cell division forms into two cells over there. The one that is going to form a self-renewing cell. This is in a memory cell that has to be left out. And the other cell is going to differentiate into asymmetrical cell division. So here also they are going to form a self-renewing cell or the memory cell, and this cell is going to form into the next cell, that is a progenitor cell. The progenitor cell is going to go and differentiate into terminal differentiation, that is the unipotent stem cell, that is a terminal or the nullipotent, where it has come to the final stage cell that it is going to be dividing. Okay, so coming to embryonic stem cells. Okay, till here we were talking about the different types of stem cells. Any doubts over there, Ma?
No,
Any
ma'am.
doubts in this? Yeah. So shall we continue with the next topic?
Yes, ma'am.
Yeah. So here we are going to talk about the embryonic stem cells. So what exactly we are going to talk about over here, and how we are going to extract. So till now what we were talking about is, if they are going to ask you to discuss about different types of stem cells, you are going to talk about all those that we are talking about. That is a totipotent, the pluripotent, the multipotent, the oligopotent, and the unipotent stem cells. So you're going to give a complete picture of what all these cells are, possibly with good diagrams. You can be able to discuss over there. Now when they are going to ask you to just discuss embryonic stem cells, so we are going to talk about that in detail over here. So this is what you're going to talk about when they ask you to write about what is embryonic stem cells and its uses. So you can see in this picture over there, the fertilized egg is going to develop into the next stage, that is the blastocyst over there. So this is the cell that is going to form into the embryo, and this is the cell that is going to form the trophectoderm. This trophectoderm is the one that is going to form the extra embryonic stem cell, that is the placenta over there. And these are the cells that is going to form the embryonic stem cells. So the scientists are interested only in these cells. So from these cells, they are going to take out, isolate this cell, and they are going to grow them in vitro for different types of cells that they want to grow in the laboratory. So here, when you're going to talk about the stages of the embryogenesis, so day one, the fertilized egg over there, and in day two, you have a 2-cell, and in day three, you have a multi-cell stages. And this is the stage where you will be calling them as uh, totipotent stem cell. So this is day 3 to day 4 is a totipotent stem cell, and after the fourth day, it goes into the fifth day. So in the fifth to sixth day, you have the blastocyst stage. So in the blastocyst stage, this is a stage, the around them, this is a trophectoderm, and the inner cell mass is called the blastocyst, or the embryonic cells that is going to form into an embryo over there. And later on, from day 11 and 14, tissue differentiation occur. So if you remember the stage over there, when we were talking about the chick embryo, the primitive streak and all those starts to form. And the primitive streak is the starting stage of where the neural gastrulation, that is the neural groove, is going to form for the development of the brain, and also for the development of different organs that has to be formed in the future. So coming to embryonic stem cells, so as we talked, these are the pluripotent stem cells, which can be isolated from the inner cell mass over there. If you can see in this picture, the inner cell mass. The unique feature of this E cells are that they distinguish them from other types of organ-specific stem cells, including the ability to maintain and expand the cells in culture for an extended time, while maintaining their normal karyotype and pluripotent nature. So they are able to maintain the karyotype, normal karyotype, if you remember the karyotype that you're talking about, and also they are able to maintain the nature of pluripotency, that is developing into various different kind of cells. So what is the origin? From where can you take this embryonic stem cells? When you're talking about a human embryo, so human embryonic stem cells are predominantly isolated from the blastocyst stage of the embryo, derived from the in vitro fertilized eggs. So blastocyst stages from day 4 or day 5, you can take out this fertilized cells, that is the embryonic cells. The primate and the human blastocyst consists of their surrounding trophectoderm. So they have a trophectoderm epithelium and an inner cell mass, and of course the extra embryonic endoderm also is formed around them. Now, if you can see in this picture, so this is the trophectoderm that I was talking about, which is going to form the future placenta, and this is the inner cell mass that is going to form the future embryo, and they are going to form different cells, that is the blastocyst stage, which is also a pluripotent that can give rise to any different type of cell. So what is the day of this embryonic? In mouse, this stage you can get at 3.5 days, and when you're going to talk about humans, in the humans it is day 5 you can get the cells. So the trophectoderm participates in the placenta and the inner cell wall mass exclusively, giving rise to all the cells of the body, whereas the extra embryonic endoderm may provide the embryo with nutrients and patterning information. How they have to form the patterns over there is what the extra embryonic endoderm is going to give information. The pluripotent stem cells are cells that are able to self-renew by dividing and developing into 3 primary parts of the cells, that's the ecto, meso, and the endoderm, that make up the human body. So there are different types of pluripotent stem cells. I want you all to have some knowledge about this and also concentrate on these points over there, because it's going to be a little bit tricky, but also very informative over here. What are the types of pluripotent stem cells? In case you are asked what are the different types of pluripotent stem cells, you'll be able to answer this when you're understanding these points over there. So these are the different types of pluripotent stem cells, that are the induced pluripotent stem cells and embryonic stem cells. So induced pluripotent stem cells, you know what we are going to talk about. In the embryonic stem cell, we are again going to talk about 3 different stem cells, that is true embryonic stem cells from the embryo, and the embryonic stem cell from the somatic cell nuclear transfer, and the embryonic stem cells from the unfertilized eggs, that is the parthenogenesis that we are going to talk about over there. So in induced pluripotent, so as the name suggests, the IPS are the outcome of the scientific research wherein normal body cell line skin cells, it is not just skin cell, you can take out any cell and you're genetically programming that to function like pluripotent stem cells, that is you're going to induce them. And this valuable medical tool for the effective treatment of some diseases condition and understanding the disease state. IPS is also derived from the adult somatic stem cells, and being the patient's own blood cells, IPS cells do not pose a problem of incompatibility leading to tissue rejection or transplantation. So this is one big major problem that scientists and the doctors encounter, that is when they take out transplantation is done. For example, they are going to transplant a heart, they are going to transplant a lung or a liver or a kidney. After some days, although the patient is recuperating very fast, but at some point of time, the tissue is not accepted. The tissue becomes a foreign body for our own cell, and they start to reject it, and then sometimes a patient may die. So when you're talking about the induced pluripotent stem cell, you're taking your own cells, you're inducing them to form that particular tissue or organ, and then you're going to transplant it. So there is no question of tissue rejection, because your body knows your own cells. So that is the importance of induced pluripotent stem cells. So this is how they can grow them in the laboratory. That is how you're going to do it. So this is a tissue from a patient, the first one, the tissue from the patient, the somatic tissue that they have taken out. They're going to isolate it and they're going to grow them. This is the second one, that is they are going to transduce the stem cells associated genes in the cells by viral vectors. So from the viral vectors, they are going to form into what type of a cell they are you particularly want them to grow. Then in the third one, they are going to harvest and culture the cells according to ES cells culture, and using mitotically inactivated feeder cells. And then what you're going to do, a small subset of the transfected cells becomes the ips cells, and this ips cells can generate embryonic stem cell like colonies. So you have to do up till this stage over there, the fourth stage over there, so that you can grow the cells. So here inside you can see the patient cells are there, and here you have the around the red color one is the one that you have induced them to become what type of a cell you want them to induce. So once the cells have accepted that viral vector genes, they're going to form by mitotically, they're going to divide and they're going to give rise to embryonic stem cells over there. So that is induced pluripotent stem cells. Now coming to embryonic stem cells, what I was talking about is this embryonic stem cells. We are going to talk about these three different types of cells in the embryonic stem cells. So under embryonic stem cells, actual embryonic stem cells from the embryo is what you're going to talk about. That is the embryonic stem cells derived from the embryo as categorized as true embryonic stem cells. So how you're going to do this? So there are a lot of in vitro fertilizations occurring, the IVF, when you're going to talk about. Not all egg cells that have been fertilized are going to be introduced into the into the patient who are going to be going with this type of IVF kind of fertilization. There are some eggs that have been left out, which is of no use for the for that time being. So all these fertilized eggs, they are given to the researchers. So there they can use this fertilized eggs, and from that fertilized egg, they're able to take out that embryonic stem cells. That's a true embryonic stem cells that we are talking about. So these are primarily utilized in the research, and also different types of work where you want to do some research works where you really use these embryonic stem cells. So these are usually obtained from discarded embryos that are formed from in vitro fertilization. That is true embryo. Now coming to the next one, that is the embryonic stem cell from the somatic cell nuclear transfer. Understand the word somatic cell nuclear transfer. So what is this nuclear transfer here? The somatic cell nuclear transfer means the process of transferring the nucleus of a somatic cell, like a skin cell, to another cell, like the egg. This results in the formation of a genetically reprogrammed cell that possesses pluripotency. So the development of such pluripotent stem cells has been successfully carried out in the lower animals. So what I was talking about, this is a picture I want you all to understand over here. So this is the somatic cell nuclear transfer. That is, you have taken a somatic cell from any cell from the body. It can be a skin, it can be a brain cell, it can be a liver cell, whatever cell you want to, you have taken it out. But easily available is our skin cell. You're going to take out the skin cell, and you're going to remove the nucleus from the skin cell. Yeah, so this nucleus is removed and kept ready. Now another cell, that is the egg cell, is taken out. The egg cell has been removed out of its nucleus. So you're going to remove the nucleus of the egg cell, and into this egg cell, you're going to introduce this somatic nucleus. So you're going to fuse the somatic nucleus into the egg cell, and that forms the clone. When you're talking about the cloning of different types of cells over there, the cloning is done in this type. That is, you're going to get a clone or a Xerox copy of the adult. So that whatever the stem cell nucleus is, that is what you're going to get. So when I'm going to take out a nucleus from my cell, and you're going to introduce my cell into an egg cell, and when it is going to give rise to a cell, that's going to be an exact copy of mine. So that's called as a cloning or a Xerox copy. Exactly, a Xerox copy is what I was talking about in somatic cell nuclear transfer. So in the reproductive cloning, you can either introduce the cell into the surrogate mother for development of a new organism, or when you're going to talk about the therapeutic cloning, you're going to culture them as a tissue culture. So whatever the tissue that you want to culture, say you want to culture a cell, a skin, you want to culture a liver, you want to culture a kidney or a bladder, you can do so in this type of somatic cell nuclear transfer. So that is what is the second one over here. embryonic stem cell from somatic cell nuclear transfer. So here there will be no problem of taking out the embryonic cells. And the next one, the third one, is the uh, embryonic stem cell from unfertilized egg. So what is this unfertilized eggs? So unfertilized eggs can be induced to form embryos without fusing them with the sperms, and the process is known as parthenogenesis. So we must have talked about parthenogenesis in various types over there. For example, when you're talking about the honeybees and sometimes even in the Drosophila, the ants, you will be talking about parthenogenesis, where the cell doesn't have to undergo fertilization. So such embryos are cultured in the laboratories for days, and then the embryonic stem cells are harvested from these cells over there. So that is embryonic stem cell from unfertilized eggs. So again, there will be no problem of we trying to take out the cell from the embryo over there. So that was the different types of cells that we were talking about. Then coming to human embryonic stem cell research. So there you can talk about what is embryonic stem cell and what are the different types of stem cells. So now we'll be talking about where all you can apply this embryonic stem cells, human embryonic stem cells, where all you can apply in the research or in regenerative medicine. So it was in 1998 researchers first time reported the establishment of human embryonic stem cell lines, and these cells existed indefinitely in the culture and are capable of giving rise to any human cell type. So where are the applications that you can do? Correction of disorders with loss of normal cells, that is diabetes. Then where insulin intolerance is occurring in the diabetes over there, so you can induce your cells to produce insulin. Alzheimer's cells, where the memory cells is at a loss, there is a memory loss over there, so you can also try to create some cells over there so that Alzheimer's is again revoked back. And Parkinson disease, as you all know, that's a complete shaking. That is when there is a connection of nerve to nerve without the stopping. If you remember the acetylcholinesterase, the neurotransmitters, when there is a continuous supply of neurotransmitters from one cell to another cell, there's a continuous shock that is occurring, and there's a continuous shake in the body. So that's a Parkinson disease. There you can also use this embryonic stem cells to cut short the connections and give rise to a better neurotransmitters, where once the information is passed, the neurotransmitters are stopped, so that there is no continuous supply of neurotransmitters, where the shakiness stops over there. That's Parkinson's disease. So this can be achieved by appropriate cell therapy. Then you have an engineering and replacement of various tissues, and for the discovery of new drugs and testing safety of drugs, and to study the development of human cells in different types over there. Okay, so that was about applications. What are the limitations that you're going to talk about? So though sometimes science research looks so easy for us, yeah, now we have got some medicines to treat all these problems. Definitely they will have some limitations, because everything has an advantage and a disadvantage. So what is the limitations of the human embryonic stem cells? So there are several practical and technical difficulties associated with the ES cell culture and the utility. It is recently in 2004 found that contamination with a medium, the most commonly used being animal derived media, this created immunological complications when such cells are utilized for therapeutic purposes in the humans. And this contamination is identified to be due to non-human molecule, namely N-glyconyl neuraminic acid. N-glyconyl neuraminic acid, because this is a non-human molecule which is found only in the animals and not in the humans. So it is suggested that for the human embryonic stem cells, products of animal origin of any kind should not be used. Why? Because when you're going to use this media for the growth of our cells, in that media you have this non-human molecule, that is the N-glyconyl neuraminic acid, that is going to get attached to the cells over there, and these are the ones that are unable to be digested, or you cannot use them, or there is no coding you don't have an mRNA to decode that information over there. So there will be a problem over there to how to do this biochemical activities in that. So that's the reason why the contamination has to be registered and they have to be addressed over there. So, an ideal alternative is to use human embryo derived connective tissue cells as the feeder layer in the embryonic stem cell. So, what can be used other than the animal derived media over there? So, how does mouse ESC stay undifferentiated? So, how? So that is one topic that we were talking about, limitations. Here now we are going to talk about the mouse. How is the mouse going to stay undifferentiated? So this undifferentiated state is peculiar in a variety of manners, as it is they require the cell suppressor possible differentiation pathway. That is, cell suppressors. There are some biochemical pathways wherein one cell is going to be differentiated into different types, but there is some repressors or the suppressors will suppress the cell from not dividing further. So that's what is called as cell suppressors. The possible differentiation pathway at the same time keeping them primed and alert so that one or the other should not be activated at a short notice. And it's a delicate balance that may easily be tipped in one direction or the another. So using exogenous as well as endogenous means of activation, for example, increasing the expression levels of OCT4, that is the octamer binding transcription factor, one of the essential stemness proteins of 50% in murine embryonic stem cell, induces the differentiation into extra embryonic endoderm and a mesoderm, while a 50% decrease in the level of expression of OCT4 would result in the differentiation into trophectoderm. So, are you able to understand? So, there is a delicate balance. It can either become this or that. That is 50% more of OCT4. This is the transcription binding factor OCT4. A 50% more, then it is going to become into an murine embryonic stem cell. That is, it can either form an endoderm and a mesoderm. But a 50% decrease in this particular OCT4 will lead into differentiation of that same cell into trophectoderm. What is trophectoderm? It is the extra embryonic cells that is going to form. And when you're talking about the extra embryonic endoderm and the mesoderm, you're going to talk about the endodermal cells and the mesodermal cells. So OCT4 is going to work on those points over there. So in the case of mouse embryo stem cells, the key external factors that maintain their undifferentiated state in the cytokine leukemia inhibitory factor. So what is the one that is going to control this OCT4? It is a cytokine leukemia inhibitory factor, that is LIF. So removal of LIF causes the embryonic stem cells to exit their pluripotent state and differentiate into more restricted precursors. So they're going to differentiate into different precursor levels. So the cytokine LIF is the one that is going to control them either to become this or that over there. So that is how mouse embryonic stem cells stays undifferentiated. So when you're going to talk about this culture, so this is again the culture that I was talking about over here, the zygote into the morula into the inner cell mass. In the inner cell mass, this is OCT4 positive. So this is the one that is going to either form the inner cell mass or the outer trophoblast over there, OCT4 negative. So when this is cultured in the outside in vitro, when you're giving OCT4, this is going to form into differentiated cell and OCT4 is negative over there. So directed differentiation. So what is this directed differentiation over there? So directed differentiation is where you're going to direct them to form into what type you want to form them. So if you can see in this picture, the inner cell mass of the blastocyst, which is an undifferentiated embryonic stem cells, when you're going to culture them, you're going to induce them, and you want to induce them in such a way they become into either the directed. So you want more amount of neuronal cells. So you're going to add that particular growth factors and you're going to get neuronal cells. No, you want pancreatic cells. So you're going to add the growth factors for the pancreas, and then from that same undifferentiated cells, you're going to get insulin secreting pancreatic islet cells. So whatever cells you want them to is called as directed differentiation. So that is a bioengineering methodology at the interface of stem cell biology and developmental biology and tissue engineering. So all this comes into play. That is, developmental biology, stem cell biology, and tissue engineering comes into play when you're going to talk about differentiated, that is directed differentiation. So this is primarily applied to pluripotent stem cells of mammalian origin, in particular mouse and human cells, for biomedical research and applications. So this method consists of exposing the cell cells to specific signaling pathway, wherein you're going to mimic them to form whatever type of cells you want them to grow. So that was about embryonic stem cells. So the different, from where you can take out the embryonic stem cells, and where how you are going to grow them, what is this OCT4, how much of 50% increase or 50% decrease will lead to what type of cell you want to. And also we were talking about directed differentiation, that is what type of cell you want them to grow them with that particular type of growth factors, you get that different type of cells. Now, coming to the next one, you're going to talk about adult stem cells. In adult stem cells, remember, we are going to talk about two types, that is induced pluripotent stem cells and, of course, the somatic cells. So your adult stem cells are very few in number. You will have to isolate them very carefully by adding some stains, because you don't have more number of stem cells in adult when compared to embryonic stem cells. But of course, nevertheless, if you're going to isolate that particular stem cell, you're able to grow whatever cell that you want, whether you want to grow a heart, whether you want to grow a brain cell, whether you're going to grow muscles or blood or skin grafting, and all these things can be done in adult stem cells. So, what is adult stem cells? So, these are undifferentiated cells found throughout the body after development that multiply by cell division to replenish dying cells and regenerate damaged tissues, also known as somatic stem cells. They can be found in juvenile, adult animals, and of course, humans. So, if you remember, when you're talking about regeneration of the damaged tissues, so here, when you have a wound or when you have a fracture, our cells, the adult stem cells that are present, they are activated, they are being induced to form that particular cells over there. So that is how we are able to repair our damaged tissue, repair the wound wherever is possible, repair the fractures, that is a minute growth of the bones over there for attachment over there, that is being repaired. You can also repair the DNAs, because during mutations or during excess UV radiation exposure, your DNA can be damaged, or they can also copy a different type. So all these can be repaired by this stem cells. So that is what is called as either dying cells or regenerating the damaged tissues over there. So the main function of the adult stem cells are to replace the cells that are at the risk of the possibility of dying as a result of disease or injury, and to maintain the state of homeostasis within the cell. So what is the function? To replace physiologically lost cell, example blood, because blood we are going to lose them every 120 days. So you need a new cells that is being. So as we are sitting and listening to the lecture over here, we have lost so many RBCs, and at the same time we have reproduced more number of RBCs over there. So a homeostasis maintained when there is a loss. The same time replenishment is occurring over there. So in that example is the blood. The GIT epithelial cells, that is a gastrointestinal epithelial cells, because the cells, the internal cell lining can be damaged frequently, because it depends on what type of food that we are eating. It can be a highly acidic food, or it can also be a highly hot food, something like paprikas, the hot mirchi that we are talking about. The epithelial cells can get degraded over there, and this has to be repaired time and again. Otherwise, it leads to different types of problems in the GI tract over there. Then, of course, the skin and the hair, because we have to mold, the old skin is lost and a new skin has to be replaced. The hair loses. On an average, we tend to lose 100 hair strands every day. All those hair strands have to be replaced every day. So, this adult stem cells come into use over there. So to divide in response to tissue injuries and infections, so for example, in skeletal muscles, regeneration as a response to exercise or injury. So for example, if you've done a lot of exercise or injury, you have a muscle tear, you have a ligament tear, or there is a muscular catch where you're not able to bend or move. Then after some days, you are fine. You are back to your normal self. And how is all this done? It's because of the adult stem cells that we have in our body to take care of all those cells. So, what are the list of adult tissue reported to contain the stem cells? So, for example, when you're talking about adult stem cells, you're going to talk about the bone marrow, you're going to talk about the peripheral blood stem cells, you're going to talk about the neuronal stem cells, that is from the olfactory bulb and the spinal cord. Olfactory is from the nose, you can take out some cells, so you can grow the neural stem cells up there. Then you're going to talk about the muscle stem cells, the liver stem cells, pancreatic, renal, corneal, limbal stem cells. Corneal limbal is the cells from the cornea. You can take out. There are some small cells which acts as an adult stem cells over there, and of course the dental pulp, which has those stem cells. That is the reason why we are able to maintain our dental well-being over there by taking care of them. So dental pulp. So these are places where you can take out the adult stem cells. But mostly when you're talking about what are the different types of stem cells that you find in adult stem cells, or from where you can get adult stem cells, you're going to talk about the bone marrow first, then you're going to talk about the peripheral blood stem cells, and also you can going to talk about the umbilical cord stem cells. So these are the three places from where you can definitely take out this adult stem cells. So these adult stem cells are undifferentiated cells that reside among differentiated cells in a tissue or an organ, and they have the ability to renew themselves and differentiate it into specialized cell types. So while embryonic stem cell can become all cell types, adult stem cells are limited to differentiated into distinct cell types. That is, they are either multipotent or unipotent stem cells. So what is a multipotent stem cell? So this is the ability to generate a progeny of several distinct cell types. So for example, glial cells or neurons. So you know what a glial cells is? That's a it's not a neurons, but it's a helper cells to take care of the neurons of this. So those are called as glial cells. As opposed to unipotency, which in term is a term for cells that are restricted to producing a single type of cell, which is formed as only one type, that is unipotent stem cells. The primary roles are to maintain and repair the tissue in which they reside. Adult stem cells are rare and generally small in number, but they can be found in number of various tissues of adult organisms. So remember, they're very rare, they're very small amount, but definitely they are found in adult cells. The only thing is you have to find out where it is, because it is a little bit difficult to find out them. Plasticity. This is one more important topic when you're talking about adult stem cells. Plasticity. What do you mean by plasticity? Here we are not talking about plastic. So here plasticity has a different meaning in the stem cells. So the term plasticity means that a stem cell from the adult tissue can generate the differentiated cell types of another tissue. That is, when one cell is destined to form into one cell, you can induce them to form another type of a tissue. That's called as plasticity. So discoveries in recent years have suggested that the adult stem cells might have the ability to differentiate into cell types from different germ layers. Different germ layers. That is, germ layers is I'm talking about here ectoderm, mesoderm, or endoderm over there. So for instance, neural stem cells from the brain, which are derived from the ectoderm, can differentiate into ectoderm, mesoderm, and endoderm. That's called as plasticity. So remember, neurons, the nervous system comes from the ectoderm. But this stem cell, the neural stem cells, can also be differentiated into other different types of germ layers. That is called as plasticity over here. The stem cells from the bone marrow, which is derived from the mesoderm, can differentiate into lung, liver, GI tract, and also skin, which are derived from the endoderm and the ectoderm. And this phenomena is referred to as transdifferentiation or plasticity. That is, one germ layer forming into another different types of organs from different types of germ layer is called plasticity. So it can be induced by modifying growth medium. So for everything, you have to remember that it needs a growth medium. That particular medium that you need them to grow into is called growth medium. When stem cells are cultured in vitro or transplanting them to an organ of body different from one they were originally isolated from. So the more recent finding that pluripotent stem cells may reside in blood and adult tissue is in a dormant state. So what they are thinking is the pluripotent stem cells are still there somewhere in a blood or in adult tissue, but in a dormant state. Dormant state is they are not going to differentiate themselves, or they are in a state of rest. So
Yes.
they will be waiting when the body needs, during emergency they will express themselves. But we are still in a nascent stage. There are still research that is going on how to find out this pluripotent stem cells in the adult stem cells. So what are the different types of ER? Hematopoietic stem cells, that is adult stem cells of ER. Hematopoietic stem cells is the first one. These are the cells that can differentiate into blood cells, and the process is called as hematopoiesis. And these are found in the bone and umbilical cord. And they are generally dormant when found in adult due to their nature. Mammary stem cells. So these are mammary stem cells, that is the mammary glands. They provide source for the growth of the mammary glands during puberty and gestation, and plays an important role in carcinogenesis of the breast. So this is one problematic over there, where we have a lot of cancer cells also present in the mammary glands, and that's the reason why we have the breast cancers. So of course, with our immune system, we are able to control those carcinogenic cells over there. So mammary stem cells have been isolated from the human and the mouse tissue, as well as from the cell lines derived from the mammary glands. So intestinal cell stem cells, intestinal. So we have stem cells even in the intestines. So what are the where do you have this stem cells in the intestine? It's at the crypts of lubricants. So if you can see over here, so this is a lubricant crypts over here. So these are the places where you have this stem cells. So what are they? The intestinal stem cells divide continuously throughout life and use a complex genetic program to produce the cell lining the surface of the small and the large intestine. Intestinal stem line resides near the base of the stem cells called the niche, or called as the crypts of lubricants. So if I'm going to take out one villi over there and I'm going to show it over here. So these are the differentiating cells, and this part is a transit amplifying cell, and this cells in the pink, the purple and the blue color cells over there. So these cells are called as the crypts of lubricant, and these are the intestinal stem cells, because intestine have to always be lubricated. Otherwise what happens, they can get degraded, because it is continuously in the process of digestion different types of food, various types of food, because we are always taking different types of food. It can be acidic, basic, or alkaline. So they have to maintain the cell structures, because sometimes the cell might die, and this crypts of lubricant where the stem cells are present, they are time and again going to replenish us with a normal type of cells. So that is a source of intestinal stem cells over there. Now coming to mesenchymal stem cells. So where are these mesenchymal stem cells formed? So these are the stromal origin, and they have been isolated from the placenta, the adipose tissue, that is the fat bodies, the lungs, the bone marrow, the blood, and the Wharton's jelly. Where's the Wharton's jelly? This is found in the umbilical cord and the teeth over there. So this is the umbilical. This is a picture of an umbilical cord, and this is a place where the Wharton's jelly is present, and this is the place where you can get the mesenchymal stem cells. And of course the teeth also has that pulp we're talking about, so they also have this mesenchymal stem cells. And these stem cells are attractive for clinical therapy due to their ability to differentiate, provide trophic support, and modulate innate immune response. So innate immune response, remember the immunology, innate immune response. And these cells have the ability to differentiate into various cell types, such as the osteoblast, chondroblast, adipocytes, neuroectodermal cells, and hepatocytes. Now remember, I was talking about when I was talking about the class, blast, sites, and the clast. Remember these 3 points over there. If you have not listened to it, please note down in your notebooks. Blast, 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 site, cytes, sites, and clast, clast, clast. So whenever there is a suffix present before these words, when you're talking about, for example, in this picture, when you're going to talk about the corn osteoblast, these are the immature cells. Blasts are immature cells. Sites, cytes, sites, are mature cells. And when I'm talking about a clast, 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 osteoclast, for example, if I'm going to talk about the osteoclast, that means the cells are on the verge of dying. So blast, immature cells; sites, mature cells; clast, the cells that are going to die. So wherever you find this, don't get confused. Just remember the last prefix, the suffix that is present, and then you will be able to identify what type of the cells that particular point is going to be talked about. The inflammatory response can be modulated by adipose derived regenerative cells, which is called as ADRCs, including mesenchymal stem cells and regulatory T lymphocytes. So, inflammatory response, when you're talking about the immune response over here, you're going to talk about the adipose derived regenerative cells and, of course, the T lymphocytes. So, here, adipocyte derived regenerative cells, this again reminds us that not always the fat bodies are bad to us, because fat bodies are also important to generate some important cells over here. So when you're talking about the regenerative cells, then also when you're going to talk about the vitamins, there are some vitamins which are soluble only in the fat. If you remember the first year when you were doing this experiments of vitamins, vitamin fat soluble vitamins. So this fat soluble vitamins can be soluble only in the presence of fat and not in the presence of water. So definitely we should have a little bit of fat in our body, which will help us to form into different types of regenerative cells and, of course, the T lymphocytes. The mesenchymal stem cells thus alter the outcome of the immune response by changing the cytokine secretion of dendritic and T cell subsets. Cytokine. Remember what do you mean by the cytokines? When you're talking about the immune cells, the cytokines are the one that is going to be secreted so that they can engulf the microorganisms or the antigens, so that they can kill them. So these are the cytokine secretions that you find in the dendritic cells and also in the T cell subsets. So this results in the shift from pro-inflammatory environment to anti-inflammatory or tolerant cell environment. So this is the one that can induce the autoimmune disorders also. So when you're talking about this particular picture over here, this is a very attractive clinical therapy when you're going to talk about the mesenchymal stem cells. So mesenchymal stem cells can be taken out from the bone. So once the cells are isolated from the bone marrow, some of the cells are going to self-renew, and some of the cells are going to be utilized for clinical therapy in epithelial cells, the neurons, the connective cartilage, fat cells, bone marrow when you're talking about mesoderm, and when you're going to talk about the endoderm, you can form them into muscle cells, gut cells, and the lung cells. So this is the importance of the mesenchymal stem cells over there. Now, when you're talking about the endothelial stem cells, so we have talked about ectoderm, we have talked about mesoderm. Now we are going to talk about the endothelial stem cells over there. So endothelial stem cells are the one of the three types of multipotent stem cells found in the bone marrow, and they are rare and controversial group with the ability to differentiate into endothelial cells and the cells that line. So for example, these are the blood capillaries, the veins and the arteries. So the cells that are lining them, these are the endothelial stem cells that you find over there. So these are the stem cells, the endothelial cells, the smooth muscles, and the outer covering of these arteries and the veins. And this is the histological sections where you can easily find the fibroblast, the adventitia cells, the media cells with the smooth muscles, and the intima consisting of the endothelial cells that are present over there. So these stem cells are important aspect in the vascular network, that is the blood vessel network, and even influencing in the motion relating to the white blood cells. How the white blood cells movement is occurring is all because of the endothelial stem cells. And they are also able to generate new blood vessels and process known as vasculogenesis. So you are going to talk about two different types, vasculogenesis and angiogenesis. In the vasculogenesis, when you're talking about vasculogenesis, we are talking about the new blood vessels, and when you're talking about the angiogenesis, we're talking about the already blood vessels that are being developed, that is the capillaries that are going to be developed, is called as angiogenesis. And in adult, new blood vessels mainly arise through angiogenesis. So from here, if you can see, hemangioblast, that is the cells that are going to form into vasculogenesis, that's the blood vessel, and the blood vessels is going to form the capillaries over there, that's called as angiogenesis. And these are the three different types of vessels that are formed due to, sometimes it is when you're going to give some good exercise, you have an healthy angiogenesis. When there is ischemia is when there is a blood clot, and the blood clot leads to loss of blood supply to that particular area, so that's called as the therapeutic angiogenesis. Over there, ischemia is formed and there is no proper capillaries being formed over here. And infarction and regenerations, sometimes unhealthy angiogenesis can occur during infections, and regeneration is how this particular problems arises over here. And all this can be treated by endothelial stem cells. And you're going to talk about, are we running short of time, or there's 5 more minutes to go? When do you have the next class, ma?
3:15.
Pardon, ma? 3:30. Okay. So I'll just continue for 5 minutes and then I'll wind up this topic. So remember, I was also talking about olfactory adult stem cells. So olfactory adult stem cells is where you can see this adult olfactory bulb over there, and this is where you can clearly take out that stem cells over there from the mucosa, human mucosa, and these are the potential for therapeutic applications where this can be harvested with ease. That is, when you're talking about old patients also, in the old patients, this is the best way of taking out the stem cells to produce their own type of cells for therapies. So this is adult olfactory stem cells that can be found over there on the lining of the nose, and these are involved in the sense of smell. Neuronal stem cells, the existence of the stem cells in the adult brain has been postulated following the discovery of the process called neurogenesis. Genesis is giving birth to new cells over there. And while lower organisms possess extensive capacity for neural regeneration, evolutionarily higher organisms, including humans, have limited their ability to regenerate nerve cells, posing significant issues for treatment of injury and diseases of the nervous system over there. So here, from a neuronal stem cell, self-renewing, they can either form the neuron, they can either form the astrocytes, or also they can form the oligodendrocytes. But in humans, as they say, the capacity to regenerate decreases over there. So where all you can find this neuronal stem cells? One is at the subventricular stem cell zone, that is the lateral ventricles of the brain. Under the lateral ventricles of the brain, you have the small brown color structures on either side. So this is where the brain stem cells can be harvested. And one more is at the hippocampus region over here. So this is a dentate gyrus, and this is the place where you can also take out this nerve cells for stem cells over there. So these are the two places where you can get these neuronal stem cells for development of different types of cells that you want them to grow. And there is also the neural crest stem cells. Remember when during the development of the primitive streak, and when the neural groove is formed, here you can see the neural crest cells that are formed over here. Now these are the cells that can again form into a proper stem cells. So that's the neural crest stem cells, which can form into different types of cells that you want them to grow. And there are different similar cells that have been found in the gastrointestinal tract, the sciatic nerve on the sciatic muscles, the cardiac outflow tract, and the spinal and the sympathetic ganglia. So these are the cells that can generate neurons, Schwann cells, myofibroblast, chondrocytes, and the melanocytes. Then you have one more type of cells, the testicular cells, that is the gonadal cells from the gonads of the male reproductive organs. You have some cells called the spermatogonial stem cells, and these are the cells that can also form into different types of cells. That is, when self-renew, they can be formed into different types of cells that can be used as stem cells over there. So the extracted stem cells are known as human adult germ line stem cells, and the multipotent stem cells have also been derived from germ cells found in the human testicles. And yeah, so if you can see in this picture, this is the development in the stem cell research that is found. This was taken out in 2004, but the patient over here, the lower limb were paralyzed after an accident in 1985, and this person had to completely use either the chair, wheelchair, or a bed, but she was not able to get up and walk on her own. But the stem cell therapy had made her to walk again. That is, this was conducted in 2004, and in just 3 weeks they found that the patient was able to walk, and with the help of a walker. So definitely stem cell also shows some advance and some promises for treatment of different types of accidents, and also regenerative medicine therapy. So when you're talking about regenerative medicine, this is again the scientist who's growing a bladder in vitro. So that's a bladder over there. We are able to grow a complete bladder in vitro with the help of a scaffold, biodegradable scaffold. What do you mean by a scaffold? The shape, whatever shape you want, that's called a scaffold. And you're going to add the stem cells over there, and the stem cells is going to grow into the shape of whatever scaffold you have given. And after that, the scaffold is removed and the organ is transplanted to whatever patient that is needing over there. So that is one more regenerative medicine that is occurring. So this was taken from a Lancet, that's a journal, Lancet, in April 2006. So potential uses of stem cells, basic research over there. So that is at a molecular mechanism for gene control and gene expression and differentiation, and of course the stem cell therapy for cancer. And under biotechnology, you can talk about safety testing for new drugs, and also the screening potential of the drugs, wherein the screening of potential drugs cannot be done in a human, so you can do that in a stem cell. So for example, cancer cell lines for anti-tumor drugs, then availability of the pluripotent stem cells for testing different types of diseases, and then cell-based therapies for Alzheimer's, spinal cord injury, stroke, severe burn, heart disease, diabetes, osteo and rheumatoid arthritis. And these can form the vehicles for genetical manipulations and therapeutic cloning, of course, and also in cancer. So we are left with hematopoietic stem cells. This we will continue in the next class, that is tomorrow. As far have you all understood whatever we were talking about, or do you have any doubts over there? Any doubts regarding the topics that we have talked about? You can
Hello,
cover.
ma'am.
Yes, ma.
Cells removed from cadaver, will they function normally for insulin production?
Uh,
Before how
in,
many hours we have to transfer it to lab?
yeah, that all is definitely a question that has to be taken care of. So here what they are talking about is gene manipulation, where they are going to take out that particular gene which is inducing the insulin production. So the gene that is particular from the cadavers, that is going to be taken out, and that gene can be copied into the vectors and then introduced over there. So that is what they were talking about in cadavers. So it's not that directly something like cornea, for example, when you're going to transplant the lens, the cornea has to, that has to be done within 3 or 4 hours of death.
2 hours, sir? Okay.
So this is a complete cadaver, he's saying. What do you mean by cadaver? It's a complete dead body, a long back dead bodies. They can take out the genes and they can manipulate with that particular gene.
Okay, madam. Thank you. Madam, can you please share this PPT?
Definitely. PPT will be shared in the group. That is, let me complete this topic, and then I will, as and when I'm completing each topic, I'll keep sharing the PPTs for you all in the group.
Okay, madam. Thank you.
Welcome. Okay, so shall we leave the meeting right now? We'll meet you tomorrow, right?
Thank you.
Thank you.
Thank you, madam.